Inside the mouth of a Neanderthal who died approximately 50,000 years ago in northern Spain, archaeologists found something that shouldn't be there. Not food, not meat, not starchy roots, not anything with nutritional value. What they found, trapped in the mineralized plaque on his teeth, were the chemical compounds of two plants, yarrow and chamomile. Both bitter, both nutritionally worthless, both well documented across cultures and centuries for their medicinal properties. This individual also had a dental abscess and an intestinal parasite, both of which cause significant pain and inflammation, both of which yarrow and chamomile have documented biochemical activity against. A Neanderthal 50,000 years ago with a toothache and a stomach infection apparently went looking for something that would help, and found it. Not from a doctor, not from a pharmacy, from a forest where the answer had been growing all along. The question is, how did he know? To understand how ancient humans navigated a world where the line between poison and medicine didn't exist the way it does for you, and what that means for the body you're running right now, we need to untangle 5 things. The specific find that changed what scientists think about ancient pharmacology, why the category poisonous plant is a modern oversimplification, how ancient humans learned which plants to use, and how the co-evolutionary relationship between your body and plant toxins that started long before any human existed, and what this means for the way modern people think about medicine, bitterness, and intensity. Let's go. Here's the find that anchors everything. In 2012, researchers from the universities of York and the Basque Country published the results of analyzing dental calculus, mineralized plaque, from 5 Neanderthal individuals excavated at El Sidrón cave in northern Spain, dated to between 47,300 and 50,600 years ago. The calculus of one young adult male contained chemical compounds specific to yarrow and chamomile. Neither plant has meaningful caloric value. Both are strongly bitter. The TAS2R38 gene, the gene for bitter taste perception, was confirmed present in the El Sidrón Neanderthals by a separate genetic study. They could taste the bitterness. They ate the plants anyway. The same individual had physical evidence of a dental abscess in his jaw and molecular evidence of intestinal parasites in his calculus. The same individual also showed traces of poplar bark, which contains salicin, the natural precursor to aspirin. He wasn't eating medicine by accident. He was choosing it. To understand how he knew what to choose, you have to dismantle a category that feels obvious, but is actually very recent. The distinction between poisonous plant and medicinal plant, between something that harms and something that heals, is not a natural division written into the plant world. It is a dosage question. Almost every plant compound that has pharmacological activity at therapeutic doses is toxic at higher doses. Almost every plant compound that is toxic at high doses has some pharmacological activity at lower ones. The 16th century physician Paracelsus articulated this most clearly. All substances are poisons. There is none which is not a poison. The right dose differentiates a poison from a remedy. What sounds like a modern insight was, in practice, the empirical framework that prehistoric humans had been running for hundreds of thousands of years before Paracelsus wrote it down. Ancient humans didn't encounter a world divided into safe plants and toxic plants. They encountered a spectrum of plants with varying intensities of effect, some of which were predictably lethal, some of which produced localized discomfort, some of which altered consciousness, some of which reduced fever, killed parasites, stopped bleeding, or suppressed pain when consumed in specific amounts through specific routes. Learning to navigate that spectrum wasn't recklessness. It was the original pharmacology. The evidence from El Sidrón fits within a much larger pattern. Archaeological analysis of Neanderthal sites across Europe, using dental calculus, pollen analysis, and plant microremains, has identified consumption of non-nutritional plants, including yarrow, chamomile, groundsel, and ephedra, at multiple sites. Paleolithic and Neolithic sites in the Levant, Morocco, and Central Asia have yielded evidence of deliberate plant use that cannot be explained by diet alone. At Shanidar Cave in Iraq, the famous site where a Neanderthal was buried with flowers, pollen analysis suggested the deliberate placement of medicinal and aromatic plants, though this interpretation remains debated. The pattern that emerges across the evidence is not a species stumbling accidentally into plant toxins. It is a species engaged in a long, empirical, trial and observation process of mapping the plant world's pharmacological landscape, learning over generations what each plant did under what conditions in what amounts. 50,000 years before clinical trials, the methodology was try it, observe, survive, tell someone. The sample sizes were small, the stakes were high, the data eventually accumulated. And here's the part that makes this more than just a story about ancient ingenuity, because the process wasn't only cultural. Your body was already deeply involved in it, at the molecular level, long before any human decided to eat anything. Here's what your tongue knows that your brain doesn't consciously process. Bitter taste is not randomly distributed across foods. It is concentrated in plant secondary metabolites, the chemical compounds plants produce not for nutrition, but for defense. Alkaloids, tannins, glucosinolates, terpenes. The same class of compounds that makes a plant poisonous at high doses makes it taste bitter at any dose. Your bitter taste receptors, you have approximately 25 distinct types, far more than your receptors for sweet, salty, or sour, evolved specifically to detect this chemical class. Not to enjoy it, to flag it, to make you pay attention. Bitter is not a flavor. Bitter is a warning system, and a very sophisticated one. Which raises an uncomfortable question. If bitterness is a warning, why did ancient humans keep eating bitter things on purpose? And why does your body still respond to them in ways that go far beyond taste? The answer starts with what happens in your body when you encounter a bitter compound, and it's not just aversion. When bitter compounds enter your digestive system, they trigger a cascade of responses that go well beyond the taste buds. Bitter receptors, TAS2R receptors, are expressed not just in your mouth, but throughout your gastrointestinal tract, in your lungs, in your immune cells, and in your brain. They are part of a surveillance system that monitors what's entering your body and responds accordingly, increasing bile production, stimulating digestive enzyme release, modulating immune activity, and in some cases, triggering the cellular machinery that clears pathogens and damaged cells. This is the biological infrastructure that ancient humans were, unknowingly, activating when they ate bitter medicinal plants. When the Sidrón Neanderthal with an abscess ate yarrow and poplar bark, he wasn't operating on mystical intuition. He was activating a body that had, over millions of years of co-evolution with the plant world, built molecular machinery specifically tuned to respond to plant secondary metabolites. This co-evolutionary relationship runs deeper than most people realize. Plants produce alkaloids and terpenes as defenses against insects, fungi, and herbivores. Herbivores, including primates, evolved metabolic enzymes and receptor systems that detect and process these compounds. In some cases, the plant compound turns out to be useful to the animal, antiparasitic, antibacterial, anti-inflammatory. In others, it's genuinely dangerous. But the relationship is dynamic. Your liver's cytochrome P450 enzyme family, which metabolizes a huge proportion of pharmaceutical drugs, evolved specifically to process plant secondary metabolites. It predates pharmaceutical chemistry by tens of millions of years. You are not a body that was designed for a clean, plant toxin-free diet that modern medicine then complicated with drugs. You are a body that co-evolved in constant chemical negotiation with the plant world and the molecular machinery your cells use to process aspirin, ibuprofen, antibiotics, and chemotherapy drugs is the same machinery that evolved to handle what your Neanderthal ancestors were eating in Spanish caves 50,000 years ago. The pharmaceutical industry didn't invent a new category of chemistry. It isolated, concentrated, and standardized compounds from a chemical relationship your body had been managing for millions of years. Willow bark contains salicin, the precursor to aspirin, and has been used across dozens of unconnected cultures for fever and pain. Foxglove contains digoxin, still used in cardiac medicine today, and was used in folk medicine for heart conditions before anyone understood why it worked. Quinine, still the basis of malaria treatment, comes from the bark of the Cinchona tree, used by indigenous Peruvians for fever centuries before European contact. Artemisinin, the most effective modern anti-malarial, comes from sweet wormwood, used in Chinese medicine for over 2,000 years. In each case, the sequence was the same. Ancient humans observed the plant's effects, learned the dose that helped rather than harmed, transmitted that knowledge across generations, and eventually handed it to pharmaceutical chemists who isolated the active compound and put it in a pill. Modern medicine did not replace ancient plant knowledge. It put it in a blister pack and wrote, "Take with food," on the label. Step back, and eating poisonous plants on purpose stops being reckless and starts being something completely different. It was pharmacology without the word for it. It was a systematic, empirical, multi-generational project of mapping the chemical landscape of the plant world, learning what each compound did, at what dose, through what route, under what conditions. The Neanderthal at Sidrón with a toothache who ate poplar bark wasn't primitive. He was applying knowledge that had been accumulated by observation, passed down through social learning, refined across generations, and eventually verified by 21st century chemical analysis. The line between poison and medicine that feels obvious to you is not a discovery. It is a convention, a simplification that collapses the real complexity into two neat categories so that modern people don't have to think about dosage. Ancient humans didn't have that simplification. They had something harder and more accurate, the actual spectrum, learned through direct experience, maintained through cultural transmission, and written into the receptor systems and metabolic enzymes of every cell in your body. The pharmacy didn't replace the forest. The pharmacy learned from it. Here's the one thing. The next time you eat something bitter, dark leafy greens, black coffee, dark chocolate, fermented foods, and feel a slight resistance in your body before it settles into something almost pleasurable, notice what's happening. Your bitter receptor system is not just detecting flavor. It is running the same ancient surveillance process it was running in prehistoric forests, flagging a plant's secondary metabolite, assessing the dose, and beginning to respond. The slight aversion followed by satisfaction that many bitter foods produce is the sensation of a very old system deciding this input is within acceptable parameters. You don't have to eat yarrow for a toothache. But the system that the Sidrón Neanderthal used to find that answer, the one that made him reach for bitter bark instead of sweet fruit when he was in pain, is still in you, still running, still paying attention. It just usually gets ignored in favor of things that don't taste like medicine. And if the idea that your body was built in a long chemical conversation with the plant world changes how you see bitterness, wait until you hear what ancient humans were doing with plants that took that conversation all the way to the edge of consciousness. Why ancient humans deliberately starved before rituals, and what it did to their brain chemistry. That one's next.